Thanks. I'm going to be following on much of what Lori's already described, so if there's a little bit of duplication, please forgive me. Additionally, some of the material that I'm going to be covering is a little abstract. I promise we'll find some way to get slides up for you so that you can review them, and if you want any additional information on our work, you can go to our website and collect some of that. As you can see from some of these titles, we're involved with many different kinds of work. I'll start at the bottom first. The Northeast Biodefense Center is a regional center for New York, New Jersey, and Connecticut, which initially focused on bioterrorism, but expanded very rapidly to consider emerging infectious diseases, because nature really is the origin of much of what we're concerned about. Additionally, as of September 30, we're the only academic collaborating center under the auspices of the World Health Organization that's specifically focused on doing diagnostics and surveillance for these types of disorders, and the Center for Infection Immunity is our umbrella organization. And I've titled this Microbe Hunting in the 21st Century because I think that's probably the most appropriate title. But in fact, I'm alluding here to Pandora's box. And as you might remember from mythology, as she opens up this box, all the furies of the world escape, and just as she's about to close it, she hears one plaintive wail from inside, and that was hope. And she lets that out as well, and hope is diagnostics, information, and science. Now, although I'll be focusing on the role of microbes as enemies, I think it's important to recognize that they're not purely that. We live in a sea of microbes. In fact, we're outnumbered by microbes, not only bacteria, but also viruses, and they are critical for us. They played important, pivotal roles in evolution. Without retroviral elements, for example, in the placenta, we would not be able to participate in live birth. Similarly, we've got data now that's increasingly persuasive to suggest that many autoimmune diseases and allergies and so forth may be a function of immunological imbalance. And microbes also have important roles in nutrition, and many of you are probably interested in probiotics, and I think that we will begin to understand much more about what we consider to be normal and abnormal microflora. Additionally, microbes have been key in genetic technology. They've given us drugs, they've given us vaccines, and many of the problems about which we've heard over the past few days could be addressed by microbes. Microbes have the ability to sequester heavy metals, toxins of various sorts. They can also be used for nanotechnology to build batteries, to facilitate computing, so they can be friendly as well. So infectious disease is really microbes and their hosts out of balance. And what I'm going to try to present to you today very rapidly is a number of technologies. I'm going to try to give you some examples of real-time applications. And the point that I'd like you to take home is that we do have the tools to address the risks of pandemics, and the implementation difficulties are really political and logistical, and Lori's alluded to those quite elegantly just a few moments ago. The other point I want to make is, although we typically think of infectious diseases as being acute, pneumonia, diarrhea, encephalitis, and such, there's increasing evidence to suggest that a wide variety of chronic diseases may have roots in infection as well. And I'll try to provide you with some examples. Now, this very busy slide from Anthony Fauci, the director of the National Institute of Allergy and Infectious Disease, tracks the appearance of infectious disease over the past decade or so. And as you'll note, the deliberately emerging one, and I wish I had a slightly better laser pointer, but I don't. If you have one, I'd appreciate it. Most of these are naturally emerging. So you can see some of the red ones, like hantavirus pulmonary syndrome, cryptosporidiosis, SARS, influenza, Nipah virus, enterovirus, and so forth. I could give you an hour lecture on each one of these, and obviously I can't. So I'm just trying to give you a sense of what's happened over the past 10 years that we know about. Now, Lori's also described for you many of the things that contribute to this. I just want to give you a sense of the scope of the problem. We are a vertebrate species. If you just consider the 50,000 vertebrate species about which we know, and you presume that these serve as a niche for each of 20 viruses, we can predict 1 million vertebrate viruses. And our current state of knowledge is such that 99% of these remain to be discovered. And each of these is a potential problem. If I had to identify one single factor that was the most important in thinking about the emerging infectious diseases that most threaten the world, it would really be agriculture. And Lori showed you those huge markets with live animals. I think that's ground zero for much of this. But you can see that over the past 30 years, we've had this explosive transition where we now share materials, foodstuffs around the world. In the United States alone, you can see evidence that a little contamination goes a long way. So when I was a kid, we would eat on one cow, maybe a couple of hundred families. Now, hundreds of cattle are put together on crowded feedlots. They're somehow ground up to make 50,000 kilogram lots of ground beef. These are shipped to hundreds of United States supermarkets. So in fact, you can see that something can spread very, very widely and have an enormous impact. I'm going to shoot myself instead of the screen. Okay, and this is, I'm not going to dwell on this except to say that this is obviously not ideal food prep. Now, in Hong Kong, just prior to the onset of SARS, this was a poster that was distributed by the Hong Kong tourism agency that many of you may remember. Hong Kong will take your breath away. Of course, they yanked that very, very quickly. And although, as I'll come to at the end, we're all very, very concerned about economic markets, as Larry Brian said last week, nature doesn't read Dow Jones. And these problems can rapidly move from this sort of hunky-dory, let's vacation in Hong Kong, to an absolutely dismal scenario. So in May of 2003, look at what happened to air traffic as a result of SARS and concern of SARS. If we have an opportunity, Lori and I were both in China during the middle of that outbreak, and we'd love to tell you a bit about our interaction and what that was like. She's already shown you the Hotel Metropole, but I can't get through these slides any more quickly, so I'll just rapidly through it just to illustrate the point that it went everywhere. Now, one of the things that's occurred really as a result of focus on many of these emerging infections is that we've begun to learn that it's not just thinking about human disease, it's also animal diseases as well. I was brought to New York after we did some work with West Nile virus outbreak in New York City in 1999, and it's important to recognize that as recently as 1999, nobody really cared about what was happening in the animal kingdom. So there was a veterinary pathologist named Tracy McNamara at the Bronx Zoo who tried to make connections to the human community and say, look, we're losing native bird species, animals in my collection are very, very sick. Nobody paid any attention to her. At the same time, people were working on the human outbreak, and we eventually came in using some novel molecular methods that allowed us to resolve this very, very quickly. But in fact, these only really came together, these two independent lines of investigation, between the 22nd and the 24th of September, when we realized we were talking about the same virus. And as a result, now we're working much more closely. People have begun to try to model the emergence of infectious diseases using historical data, considering issues of climate, crowding, and so forth. This is work by Jones, Dajak et al., which shows these hotspots where we're likely to see these agents emerging. So we and others have begun to focus, largely with some support from Google and the Gates Foundation, to some extent from the National Institutes of Health, to look at those people who are the intersections between what's out there in nature and what could come into the human community. So bats, rodents, primates, blood-sucking insects, and people who are exposed, like bushmeat hunters, butchers, and so on. And this is going to be key to understanding this. Now, when a patient presents to you with encephalitis and a classic Egyptian headdress, you're pretty clear that it's West Nile virus. But most of the time, that's not what happens. So we've tried to develop a number of very rapid diagnostic technologies. Each of them suits a particular purpose that allows you to find out what in fact is going on. And that's critical, because you need to know who's worried well, who's sick, who needs which drug, what vaccine should be ordered, and so on. The first and most straightforward system, and this is now implemented in many laboratories around the world, is something called mass-tech PCR. And I'll show you a quick film of how this works, and it's fairly simple. It takes about 6 hours to do. You have a clinical sample, it's sucked up into a pipette, you put it into a little plastic plate, it goes into this oven, which is called a thermocycler. We then put it in this device, which is getting smaller every month. A robot comes, collects that sample, it puts it into an instrument called a mass spectrometer, where everything is ionized. It's not quite as cool as the air jelly, but I did my best. And what happens is you throw these proteins off, they then attach to a detector, which then reads with a number of signals. And by using this kind of an approach, we can actually identify what's in a sputum sample, a blood sample, diarrheal sample, what have you. So in this case, it's somebody who's got influenza, this is SARS, this is parainfluenza virus, respiratory syncytial virus, and enterovirus, and so forth. And it's very inexpensive to implement. In instances where we don't succeed, and we still need to know, there's an outbreak, what have you, we typically go to another system, which is a microarray, and this is a very inexpensive sort of platform as well. I've got a movie which illustrates how this works too. And again, you see with somebody putting material onto a slide. The slides are about yay big. They put them in an oven, they wash them, and then what happens is that goes into a laser scanner which reads the signal, and you see a digital signature of all the different types of agents that might be present. These things are coded with 1 million different probes, and you can recognize bacteria, viruses, parasites, and so forth. This gets blown up, and then the whole thing gets analyzed using this system. Now, this is a computer system at the front end that's accessed by many people all over the world. And what happens is they access this browser, which is known as GreenLamp. It's just an acronym. And all those data are compounded and tells you that what you have on this slide is something that is binding to a paramyxovirus. Then what you have is the capacity to take that virus sequence, spread it out on a prototype of what a genome might look like, and then you look for every place that you see a green bar is where you would have expected to see signal in this particular test. Now, if you see something which is red, that means you anticipated signal but didn't find it. If there was something that was bioengineered deliberately, or that emerged in nature that was weird, you would start seeing differences in the anticipated signatures. So the right part might be red and the left part would be green, or vice versa. A lot of information can be culled from this sort of work. Now, when that fails, we use high-throughput sequencing. This, more than anything else that we've done over the past few years, has revolutionized our approach. And I'm going to show you an outbreak investigation that's now in action, which illustrates what we can do. A small team in a few days can replicate what used to take entire organizations. We can also inventory the entire tree of life. Now, what we do is we take clinical samples. This is brain, liver, lung. We extract the nucleic acid from that. We amplify it by the DNA by PCR, and I showed you what that little oven system looks like. We put those amplification products onto beads. Those beads then go into little wells. This is an electron micrograph which shows such a bead. It's a little microreactor. Amplification is done, and then we pass nucleotides over it, and as they bind in the appropriate site, they give off light, and that gives us sequence data. We then analyze those at the level of genes and proteins, and we get images across the tree of life. Viruses, bacteria, parasites, metazoa, and fungi. And it really does work. So I'm going to show you a couple of examples. This was work we did on honeybee colony collapse disorder. The lead on this was Diana Cox-Foster at Penn State. Many of you know about this disorder. We're just losing honeybees. Nobody really knows fully what's going on, but we think we have at least one culprit. So this is a normal frame, completely populated by these honeybees. We analyzed these using high-throughput sequencing. We found a wide variety of viruses. We found bacteria, parasites, metazoa, fungi, and so forth. We tested them all individually in smaller populations of bees, and as we analyzed those, looking for specificity, there was one that shone out as an important candidate: Israel acute paralysis virus. So then we decided to ask whether or not, in fact, this particular virus could cause disease. So we put the virus into sugar water to feed bees in a closed greenhouse. This is the normal lawn of bees. As you can see, they're completely confluent. 3 weeks after infection, they begin to drop in number. 9 weeks post-infection again. And although this may not be the only cause of CCD, it's clearly not good for bees. So now there's a double-stranded RNA drug that's being used for this purpose. Now, I told you I was going to mention chronic disease, and I'm going to do that as well, because I think this is a very, very important understudied area. As a result of the links between human papillomavirus and cervical cancer, we now have a vaccine to prevent cervical cancer. The same thing is true with hepatitis B viruses, where chronic hepatitis and hepatocarcinoma can be prevented. We have Lyme arthritis, peptic ulcer disease, a wide variety of these where there's a chronic inflammatory process which occurs early, which then triggers frequently neoplasia or organ failure. But there are a wide variety of other conditions that look very interesting as well. Cerebrovascular disease, people with poor dentition, much higher frequency of cerebrovascular disease, cardiovascular disease. A wide variety of other things: inflammatory bowel disease, pulmonary fibrosis, autism. It's a huge number, and again, I could talk about them all individually. Let me just show you a couple of animal models to illustrate this point. This is a mouse model that we've tried to establish for mental illness. In this case, we're trying to look at what the effects are if a mother saw some sort of immunological stimulus, be it a toxin or infection during pregnancy at different time points, what might it do? So what we're doing is the gestational period of a mouse is about 21 days. Halfway through gestation, this is the animal that sees the virus. What we're doing here is we measure over a period of 30 minutes the amount of time and the amount of space that the mouse covers in a cage. And we simply do this by tracking it visually. So this is a control animal wandering all over the cage. This is an animal that's seen the virus. It settles down in the corner of the cage and doesn't have anything to do thereafter. This is an animal that's infected 2/3 of the way through pregnancy. This animal is hyperactive. So the same genetic background, mouse, the same exposure, that is to say a virus, but a different timing of that exposure, which results in a profoundly different effect. If you take animals and you infect them with viruses, their ultrasonic vocalization goes awry. So this is an animal that's normal, that's not been infected with a virus. This is an animal that's infected. And if you play a speaker recording to the mom from the pup using this approach, the mother will not respond to that. Very complicated sorts of things. The last thing I'm going to mention to you is an autoimmune disorder. This is an animal that's seen streptococcus. What we're actually doing here is rediscovering something that was originally described in the 1600s and that some think might have actually been responsible for some of the Salem witch trials. Unusual responses in certain populations to streptococcus. So if we give this animal exposure to this particular bacterium, several things happen. As you see, it has these repetitive behaviors. If you look at the ability of this animal to explore its environment, it's reduced compared to the uninfected animal. If you look at the time that if you introduce another animal into the cage, this animal is antisocial. It tries to defend or escape. And if it's unable to escape, then it submits. It just crawls into a corner and becomes less dominant. So this is an immune response, and we know that because antibodies to that bacterium label specific regions in the brain. Okay, so now I promised I was going to tell you about an outbreak. This is very, very interesting, and it makes the case that we need these tools on board. So in the middle of September in Zambia, there was a hemorrhagic fever that was identified. The individual who was affected was brought to Johannesburg for care. Several people became infected thereafter. The healthcare workers, very sort of similar to SARS. We all thought it was going to be Marburg or Ebola, but it wasn't. It was tested negative in all these different hands. We eventually got the samples. It took us a week to get them, which was the pity. But within 72 hours, working in collaboration with industry, the Centers for Disease Control, our collaborating center at WHO was able to identify the agent. And it vindicates, as Kathy Roth, who's the director of this WHO laboratory network, says, collaboration is the most productive approach. So here's what we did. So this movie actually shows you what actually happened. So the high throughput sequencing was done. We looked at samples that had come to us from South Africa. This is an Excel spreadsheet that shows how we analyzed it. We simultaneously looked at fungi, bacteria, viruses, and as you'll see, there wasn't much until we got to the level of viruses. And here, it lit up these characters, these bad actors known as arenaviruses. Now, as we began to study this in more detail, this critter has 2 gene segments. We were able to rapidly look at one segment, which encoded the glycoproteins, which are responsible for attaching to host cells, nucleoprotein, the polymerase that reproduces the organism, and every place where you see these black dots was sequence that we got using new science. And in parallel, we were doing old science. And you can see quite clearly that the new science is better than the old science. And this is just an electron micrograph which shows what the virus looks like. So the next question comes up is this something that we should have picked up before? This is what's known as a phylogenetic tree. What it does is it establishes evolutionary distance in between different viruses or bacteria or people. So this is Lassa fever virus. It's clearly very different. This is another virus that you'll never hear of. This is Dandenong virus that we studied more recently, found killed people and associated with transplantation. And this is the South African virus. It's completely different. So what do we know now as a result of having that sequence data? First of all, it's a new virus. Secondly, we can develop inexpensive diagnostic tests for it, and we can also find its reservoir in nature. Did it come from a mouse? Did it come from a bat? Did it come from a vole? And lastly, because we know something about these other viruses, we have drugs that we can use to treat it, which is exactly what we're now doing. So the take-home points very quickly are, 1, the microbiome is largely uncharted territory. We've identified a number of hotspots for infectious disease emergence, and by focusing on pathogen discovery in these hotspots, we really do believe that it's going to be possible to predict and prevent the next SARS or the next HIV/AIDS. Lastly, that these new methods for microbe hunting will allow us to rapidly respond to these outbreaks. As you've seen, we can do this now in a period of 72 hours, and I think with allusion to the chronic disease, I think they're going to transform medicine and public health. And a few final quotes from the late Josh Lederberg: "The future of microbes and mankind will probably unfold as episodes of a suspense thriller that could be entitled Our Wits versus Their Genes." And this is even more favored when I close these lectures for students. In a period when Einstein was active as a professor, one of his students came to him and said, "The questions of this year's exam are the same as last year." To Einstein said, "But this year, all the answers are different." And I know the budget is bleak, but as Larry said last week, nature has her own timetable. She doesn't watch Dow Jones. So we need to do this kind of work, and we need to establish these mobile, smaller laboratories so we don't have to bring everything back to New York to characterize it. Thank you.